This video presents a novel transfer multitask learning method for bacteria biotope rel and ner task. To alleviate the data deficiency problem in domain-specific information extraction, we use BERT and pre-train it using masked language models and next sentence prediction on both general corpus and medical corpus like PubMed. In fine-tuning stage, we fine-tune the relation extraction layer and mention recognition layer designed by us on the top of BERT to extract mentions and relations simultaneously. We will introduce our work in 4 aspects, including the bacteria biotope task description, our methodology, experiment and results. Bacteria biotope BB task is an interesting information extraction task aiming at extracting knowledge about bacteria biotope from bioinformatics literature related to microorganism. Rel plus ner subtask focuses on extracting entity mentions of following types: microorganism, MI, habitat, HA, phenotype, pH, geographical, GE, and identification of the lives in relation between a habitat geographical mention and a microorganism mention, as well as the exhibits relation between a phenotype mention and a microorganism mention. Bottom parts of the model, including input representation, transformer encoder, are shared by both named entity recognition task and relation extraction task. The transformer is pre-trained using 2 novel unsupervised tasks, including masked language model and next sentence predicting on the combination of books corpus, English Wikipedia, PubMed and PubMed Central, PMC, corpus. We feed the final state of each token to the softmax classification layer over the tagging set. The conditional random field, CRF, layer takes the sequence of output score vector from the softmax classification layer. We observe that each microorganism entity may have multiple relations with entities of other 3 types. Moreover, all types of relation must contain a microorganism entity. Thus we take the microorganism entity as the center of relation prediction task. The input representation of each word in sentence consists of 3 parts: word vector, the embedding of features and positional encoding. A pre-trained word embedding using skip-gram model is used to map each word to a dense vector. The features we used are described in table. Each feature is represented by a 1-hot vector and parser feature embedding layer. Positional encoding is added to make the model capture the relative and absolute position of each token. The 3 parts are concatenated and fed into transformer encoder. Results show that each feature listed in table plays a key role. Our model suffers serious performance degradation without any one of the 4 input features. Here we introduce some external resources that we use in experiment. We use Google Word 2vec tool to train word embeddings on corpora composed of PubMed, PubMed Central corpus and English Wikipedia corpus. The LTP tool is used for sentence level dependency parsing, and the NLTK tool is used for sentence tokenization and part of speech tagging. The evaluation results show that our method achieves the best performance on all metrics, including slot error rate, precision and recall. in the bacteria biotope rel plus ner subtask. Our model achieves improvements on BB dataset comparing with the other 4 models. Particularly, our model significantly outperforms the pipeline baseline by -0.525 SER.